Good morning, everybody. All right, where are we at? Still up and running. Okay, cool beans. So essentially we ended up around here. And, just as a quick recap, while you get your brains started up and running, is gene structure was really different between prokaryotes, which are very simple, sort of, and eukaryotes, right? So prokaryotes have like a linear gene, right? The DNA sequence is transcribed directly into mRNA, and that mRNA is translated directly into protein. Bacteria do have other wrinkles, right? So bacteria tend to actually group different genes that are involved in the same process, say like a metabolic pathway or something, into one kind of large unit called an operon, right? We don't do that in eukaryotes. However, eukaryotes are very different in that they have an interrupted gene structure, right? Where the coding information, the coding regions, right? That's what these exons are interrupted by introns, and those introns need to be removed. There are also other structures which are not translated. These are involved in initiation and control of translation and mRNA stability. Right, so these are the UTRs. So they're involved in the control of translation. So that's 5' and 3', and RNA stability. That's mostly the 3', as far as I can tell. The poly A tail at the end is also involved in mRNA stability and translation. Right, so there's a lot more going on in a transcript from a eukaryotic gene in many ways than a prokaryote. And we'll come back to, how these mRNAs are processed a little bit later on. And so because of those introns, right, and there's always at least one, but sometimes there are many, right? So there are some genes which have, 10, 12, 13 introns in them, right? The actual coding region of a eukaryote gene is about the same as a prokaryote. So between, 1.5 and 3,000 base pairs in length. The actual size of a eukaryotic gene, though, can vary from not much bigger than that to being tens of thousands of base pairs long because of those introns. However, those introns, and this is something we'll get to in the control of gene expression, these introns allow different forms of that mRNA to be made, depending on which ones are cut out and how, right? So there is a benefit to having them, even if they require work to get rid of. Anybody have any questions so far on anything that we've covered recent lectures? Snarfing down some very, very delicious chocolate. Very, very delicious chocolate. Okay. On to the next one. So we're going to start with transcription. And generally speaking, transcription consists of a beginning, a middle, and an end. It's like a novel, right, in a sense. And the middle bit where mRNA is actually made is by far the least interesting part. The bit at either end, particularly the initiation of transcription, are much more complex, right? But in general, transcription is simply the process of using DNA as a template to make a complementary RNA molecule, right? And RNA differs from DNA in a bunch of different ways, right? So you already know some of these. Pardon me, right? Classic one is that in RNA, you don't have thymine, you have a uracil, right? Everybody should know that already. Also in RNA, so RNA actually stands for ribonucleic acid. And it's ribonucleic acid because it has 2 OH groups, right? Just as a glucose is, glucose because it has all of its OH groups on it. DNA is deoxyribonucleic acid. Oh, will you stop doing that? Bloody autocorrects. Pain in the butt. And it's deoxy because it has 1 OH group, oops, on the 3' carbon. That's why it's called DNA. Now, RNA is actually DNA is pretty boring, to be honest. If one were to choose a nucleic acid based on how varied it is, you would not choose DNA. DNA is pretty dull. Right. See if I can find some nice pictures. Oh, there you go. So RNA can form some really crazy shapes. So you're familiar with tRNA, right? So tRNA consists of 3 what are called stem loops, right? Because they have a stem and then a loop. And if you actually kind of model it as a structure, this is what tRNA looks like, right? So it's a very complex structure. And you can get all kinds of crazy ass stuff, right? You can get these all kind of like funky structures, right? There's actually one with, here you go, this coronavirus genome, right? So this is what the coronavirus genome actually looks like, right? Because it's an RNA virus when it kind of unfolds after infection. And so it's full of these kind of loops and balls and stuff like that, all of which are important in its function, right? Stability, how it's expressed. Actually, the coronavirus genome is really weird in some ways because it actually relies on a frame shift to translate all of it, which we'll talk about later on. So RNA is super, super varied, does a lot of different things, and it's also capable of acting as an enzyme, right? This is called a ribozyme. And so the RNAs that typically actually, a lot of RNAs that do things are enzymes. So a good example is ribosomal RNA. It's a ribozyme that catalyzes formation of peptide bonds. So when you think of a ribosome, the structure that actually makes protein, you often think that it's the protein that makes protein. It's not. It's an RNA molecule, which is pretty wild. And yet it also can store information. So RNA is actually one of the lead candidates for the first and the emergence of life. It's called the RNA world hypothesis, because RNA is the only molecule that can both store information and make itself. Not completely. They haven't made an RNA molecule that completely make itself yet, but that's a possible start for life on Earth. About, I don't know, depends on who you ask really, about 3.6 billion years ago, give or take. Okay, so transcription is carried out by RNA polymerases, right? And RNA polymerases catalyze the phosphodiester bond between the 3' OH of one RNA nucleotide and the 5' phosphate of the next one, right? So very similar to DNA, but RNA essentially. And that's really just reflected here. Here's the NTP coming in, nucleotide triphosphate. We break that bond there to form the phosphodiester bond, and it uses this base pairing to incorporate the correct one at that position, right? Ta-da, just like that. So in prokaryotes, there's only one RNA polymerase, right, that does everything. And RNA polymerases are another, and there are many, many of these examples of proteins that act as complexes. And so what I mean by that is multiple different proteins from different genes, all kind of glommed together like, I don't know what kind of, actually I'm pretty not very well up on like Transformers and various kind of Japanese manga cartoons where stuff comes together to create like a giant thing. Anyway, it's like that, whichever one of those you particularly fancy thinking of, right? And so they act as a unit, right? In this case, it's called a holoenzyme, right? So enzyme that uses other stuff and has multiple subunits in it. And each one does something different. Some of them are structural to kind of make the correct shape. Some of them are catalytic, right? So they actually do stuff. You know, there's lots and lots of different roles, and they're typically carried out by different proteins within that large complex. So in eukaryotes, there are actually 3 different RNA polymerases. RNA polymerase 2 does the bulk of the work, right? So RNA polymerase 2 synthesizes all mRNAs, as well as what are called small nuclear RNAs, which actually leads to my favorite acronym, which is pronounced SNRP. We'll talk about that later in RNA processing. The other 2, one of them makes ribosomal RNA, the other one makes tRNA and the small subunit of ribosomal RNA, because there's I think at least 2 rRNA molecules in a ribosome. And when you actually model RNA polymerase, so this is RNA polymerase 2, you can see how the structure informs its function. Oh, that's my phone. So the idea here, and so this is very similar to, if you remember DNA replication, right? The proteins that initiate DNA replication wrap the DNA around themselves, and in doing so kind of bend the DNA, right, in a way that pops it open. It's like undoing a Kinder egg or something like that, to make that double-stranded, which then leads to the replication bubble. So RNA polymerases do the same, but on a like much smaller scale. because all they need is a little bit of single-stranded DNA together the template to make the RNA, but they do it in the same way. And so what you can see here is DNA coming in from the right, and it basically hits what they call this wall of amino acids, right? So it's forced to bend very sharply. And in bending, it's like kind of taking a multi-stranded wire rope and kind of bending it sharply. What you see is that the strands kind of splay apart, right? Because they can't maintain that twist around a sharp angle. And so RNA polymerase makes the DNA bend, and in doing so, the DNA pops open just for that little bit inside the RNA polymerase. And that exposes the template strand, right, which is used to make the RNA. And then our NTPs, our A's, C's, U's, and G's, come in like so, and they get added by base pairing to the 3' end. So you have, for a very short stretch of DNA, this DNA RNA hybrid, right? But as the DNA comes out of the RNA polymerase, it kind of twists back into shape, and the mRNA gets kind of fed off out the exit. Right, so this is like the general overview of transcription as it happens, essentially. Oh, and I've got a video too. Don't think I've shown you this one. This is a new edition. So every time I teach this course, I always add a little something here and there. Boop-a-doop-a-doo. I don't generally rely on videos much, but sometimes they're really useful for kind of showing stuff. Get it? So kind of the 2 points of this video is that, one, the actual transcription part is super uninteresting. Well, it's obviously very, very important, because otherwise you wouldn't get mRNA, right? Or any other kind of RNA. But in terms of kind of complexity or like what's going on, the actual transcription part of transcription is relatively simple. The initiation of transcription, however, is not. And this is actually where things get kind of gonzo crazy, which is what the 4th section of the course is all about. And so what you can see here, so the other point of this film is to really show you the different, all the different proteins, right? We're not going to learn which ones they are just yet, but we're going to at least see them in action. They're involved in starting transcription, right? And so this is a video from, well, it's not a video from a eukaryotic cell, obviously, right? Because you don't have video cameras that small. But this is a modeling of initiation of transcription in a eukaryotic cell. So you'll see a whole bunch of proteins assemble here, only a few of which are actually involved in transcription, right? So most of these involved in bringing RNA polymerase in and sticking it at the right place. And then the rest are involved in activating that RNA polymerase. So you can see these are transcription factors. That cyan thing is the RNA polymerase, right, which you can see kind of tucked behind there. And then we've got a whole bunch of other proteins called transcription factors, one called mediator. And then finally, the DNA is actually bent, and there's another protein or proteins which basically go bonk and touch the RNA polymerase, drink, and set it off on its way. I like to think of it as like a kick up the posterior. But these are, so without that activated protein, this whole complex will just sit there. Sometimes RNA polymerase may make its own way out off its own initiative, but most times that complex will just sit there waiting to be activated, which you can see here, touches, and putoink, out it goes, off for the races. Zoom. It's kind of fun to watch. Always sounds a little bit like a roller coaster at SeaWorld. So one thing you can see here is that, well, you can see that bend, right? That almost acute bend. And you can see that little patch of single-stranded DNA, right? That little bubble that's been opened. All these yellow bits are just the RNA nucleotides coming in, and this is the RNA molecule coming out. That's pretty much it. So it's a fun little video. But as you can see, there's nothing much actually goes on during the actual transcription part. That is relatively simple. So one thing to just again, like give you a heads up, which I try and do, as you've probably noticed throughout the course. Transcription in genes in eukaryotes are by default off. Right, so eukaryotic genes have to be turned on. That's part of what these proteins do, right, is to actually say, you, this one's going on, come over here. Genes in prokaryotes are on by default and have to be turned off. And so that leads to some very marked differences in how they're controlled. So just keep that at the back of your mind as we move through this section. Okay, so let's get back here. Actually, let's go even further. Here we go. So, oh, hang on a sec. Cynthia, you might need to explain that a little bit. What's LAC-PORC-S? No problem. I just want to know what it is you're asking me so I can answer it better. Something to do with the lac operon? Oh, was that autocorrect? It's like operons? What's that? Let's call it pork instead. Yeah, so the lac operon is one of the examples that we use to look at control of gene expression in prokaryotes. But we'll get to that in the next section. I'm not going to worry too much about that now. Yeah, that autocorrect is a real pain in the backside. It's kind of useful in some ways and kind of not useful at all in other ways. So now what we're going to talk about is promoters, right? So essentially in a gene, you have two regions, right? One region encodes the gene or codes the protein or whatever the functional molecule is that's produced from it. I'm going to time this down. Fucking spell. So that's the protein or RNA coding if it's a non-protein coding gene. And that is after the transcription start site. So you can see that both here, that little green arrow, and you can see it here too. And so the second part of a gene is the regulator region that controls expression. And that's typically called the promoter. There are other different kind of words involved in that, which we'll get into later. But this is the region that doesn't encode protein. It controls the binding of RNA polymerase and the activation of transcription. So in bacteria, it's relatively simple, right? And that kind of ties into bacterial gene expression always being on unless it's turned off, right? So in bacteria, that promoter largely just consists of a region of DNA that binds the sigma factor and the RNA polymerase. So 2 is relatively simple in prokaryotes. There's other stuff involved here, which we'll talk about later on in the semester. So it's not quite as simple as this image shows, but relative to eukaryotes, it's fairly simple. 2 is much more complicated in eukaryotes, if I can spell that right. I'm not going to be sad at all when I don't have to type out eukaryote ever again, I have to say. It's not a lot of fun. Now you have a core promoter, RNA pol binding, and a regulatory promoter. And that's basically fancy stuff. And so both bacteria and eukaryotes have this core promoter, right? Which is where RNA polymerase binds, a bunch of other transcription factors, this, that, and the other, right? And so in prokaryotes, it's relatively straightforward, right? You have a promoter, and so another thing to really kind of stress here is that all of these proteins recognize sequences in the DNA. And so that promoter, even though it doesn't encode a protein, it has very specific DNA sequences in it, which are recognized by proteins. And so those proteins go, ooh, there's the right sequence. And somehow, I don't actually understand fully how, they can see that sequence and stick to it. And it's usually very specific. You know, there's some kind of wiggle room, which varies from protein to protein, but they can be very, very specific in their recognition. And so if you look at bacterial promoters, you have these 2 regions, which are called the -10 and the -35 sequence. As you can see with the -10 sequence, there's lots of A's and T's here, right? This is the part of the DNA that's popped open initially to create that little bubble, right? Remember talking about this before in terms of relative strength, right, of DNA, of the bonds holding the 2 strands together. Same deal in DNA replication. More likely to find A's and T's at the initiation or replication origin. So these are called consensus sequences. And so a consensus sequence equals basically the bases that are found most commonly in each position, right? So you can see some of these differ quite widely, right? These are all different genes from E. coli. But others are very, very similar, right? So for example, REC-A, which is involved in recombination and DNA repair, has a consensus -10 consensus sequence exactly the same as the consensus. Others less so. And so that degree of similarity equals promoter strength. And promoter strength is basically how often that gene is transcribed. And that depends on the function of the product that that gene is used to make. So for example, and this is getting back to the lac operon, which we'll study later. LAC-I is an inhibitor of lac operon expression. Again, don't worry too much about this. This is just as an example. And if you look at this -10 sequence, it's really CAT, GAT is not really that close. I mean, it's not that terrible, but it's not super close. And so LAC-I is a gene that needs to be transcribed kind of constantly, but at a very low level. Always has to be around, but you don't need a ton of it. You just need there to be enough for it to do its job. Right, and so the LAC-I promoter is relatively weak, particularly if you look at the -35. Now the lac operon promoter, this is what actually controls expression of the whole, suitcase full of genes involved in turning lactose into glucose. Now, when you need that, you need it. You need a lot of it, right? So the lac promoter is a very strong promoter. Actually, it used to be used a lot, not so much anymore, for expressing proteins in bacteria, for making like insulin or whatever, right? Because it was a real high level, powerful promoter. And so when you don't need the lac operon, you don't need any of it, right? That's what the control of the lac operon is all about. But when you do, you need a lot of it. So it has a very strong promoter as a result. And this is just a visualization of what a consensus sequence looks like. Basically, it's a representation of how often you find a particular base in that position. Right, so in 8 and 9, for example, you only ever find C. In other ones, you might find other bases as well. So it's just kind of a which do you find most frequently kind of deal, essentially. Now, in terms of initiation, really all you need in bacteria is a sigma factor. So a sigma factor is a transcription factor. And in this case, it helps recruit RNA pol to the promoter. Because RNA polymerase in both prokaryotes and eukaryotes is kind of dumb as a box of bricks. It doesn't know what genes it should be binding to. Or even it's not very good at recognizing like where it should be binding. It needs other proteins to help it. And so in bacteria, that's what the sigma factor does. And really, there's not much to it. Binds to the promoter with the aid of the sigma factor, opens up the double-stranded, the helix, adds nucleotides, makes RNA. That's it. Super, 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 super simple. This is almost like lulling you into a false sense of security. Because then we get to eukaryotes. And eukaryotes are not simple. I often tell this joke, but when I started my postdoc at Penn State, there's this journal club called Gene Reg, or Gene Regulation. I was like, oh, that sounds interesting. And this PhD student, the bench behind me, said, oh no, you don't want to go to that. That's like crazy, jumping down a rabbit hole, Alice in Wonderland stuff. I was like, oh, okay, I won't go to that then. Because it is just mind-bendingly complicated. Right? It really is just astonishingly complex. And I'm not going to go into anywhere near that detail here because I like my students, I don't want to, abuse or torment them, but there is a lot going on here. Right, and so for a start, eukaryotic proteins are much more complex, much larger, and less well-defined than prokaryotes. Part of that is because bacteria are a lot easier to work on. Also, part of that, there's an awful lot more going on in eukaryotes. And so, one, we only have this thing called a Tata box, right? It's called a Tata box because of the consensus sequence, TATA. Again, more T's and A's, right? Now, this can be a variable distance, right? So the gaps here, this isn't just like missing DNA. What this is here are kind of essentially gaps introduced to kind of line things up nicely. So in reality, this box here, for whatever SIAT 8C is, I don't know what that is actually kind of over here, right? That whole bit of DNA is actually shuffled to the right a little bit. But the alignment software that kind of lines things up nice and neatly introduces those gaps to allow things to actually line up and show you the shared stuff, essentially. So this Tata box is actually really important for this protein here. It's called the Tata box binding protein. The Tata box binding protein is pretty much one of the very first proteins that binds to a eukaryotic promoter, right? So one of these little blobs, not sure which one, is Tata box binding protein. And that's what recognizes this small sequence here. There's a whole bunch of other transcription factors. So you remember sigma factor is the one in bacteria. In eukaryotes, there are 6 different transcription factors. I don't know what happened to C and G. Maybe they're involved in other things. But there's a whole bunch of them. TF stands for transcription factor 2 because it's the RNA polymerase 2, and then they're just given a letter to denote each one. And so I'm going to zoom in on this a little bit, kind of control, oops, how much stuff is going on at once. So as I said before, eukaryotes have 2 regions to their promoter. They have a short core promoter, few hundred bases. This is often almost called the minimal promoter. So the core promoter, also known as the minimal promoter, is the bare minimum, hence the name, needed for gene expression. That's not to say that that little bit on its own will work super well, right? It's just the barest minimum you can get away with to actually get transcription from that gene. Right, it's like having a car which consists of a seat, 4 wheels, an engine, and a steering wheel. Oh, and a stereo, obviously. Right, nothing else, barest minimum. So in that core promoter, you've got the Tata box and the transcription factor TF2D. Those bind to that. And essentially their job is to just sit there going like that until RNA polymerase comes and goes, all right, and grabs it and sticks it on the promoter. So in a sense, TF2D and TBP, transcript Tata box binding protein, they're like scouts, right? So they go around looking for genes that need to be expressed, and they clomp onto them and they stick up a flag and basically say, all right, this one right here, come on, let's do it. And the RNA polymerase then assembles with the aid of a whole bunch of other proteins. So here are the other 5 transcription factors onto the core promoter. Without the binding of these two, this will not happen. It is sequential. This has to happen first, otherwise this doesn't happen at all. Right. And it kind of makes sense if you think about it, because in our genome we have this huge expanse of DNA, right? And at any one time, only a tiny little bit of that is being transcribed. So you need to be super clear, like this gene right here needs to be transcribed. All this other, aching expanse of DNA does not. So RNA polymerases are going to have nothing to do with all of that stuff. Another protein that's also super important is the mediator. So the mediator, as the name would suggest, mediates between the RNA pol complex and the transcriptional activator proteins. So the mediator is essentially the switch, right, which gets flicked to turn RNA pol on. All right, so we have this whole complex and it's just happily sitting there on the transcription start site, right, which is kind of covered by the RNA pol. Now we have the regulatory promoter kick in, right? And this is recognized by a whole bunch of different proteins with different purposes and different roles and different different DNA sequences that they recognize. But essentially, these proteins interact with that complex either by other proteins or by physically bending the DNA round to bring these close, and that then activates transcription. So I go, pink, off you go, which is pretty cool. And fun fact, you have 4 digits and a thumb because of one of these enhancer regions working properly in your DNA for a particular gene, right? And so the reason why you have a thumb and a pinky is because that gene is expressed here in a little limb bud, which looks very cute. And I'll just show you what that looks like somewhere. And so basically, normally, and we'll cover this in more detail when we get to it later, you have a lot of this protein called sonic hedgehog, or SHH, expressed here, right? And so initially, your limb bud, your limb bud is actually kind of here, right? Hence bud, right? Your arm doesn't exist yet. This is in development. And so normally, kind of about where your armpit is, you get this protein produced. And depending on how much of it and other weird, crazy stuff going on, depends on whether you have your pinky, which is a lot of protein, whatever finger that is, ring finger, less protein, less, none, right? That's part of what gets you your digits and your identity. Now, if you screw around with that enhancer region and other various bits and pieces, you get what are called Hemingway's cats, because of the guy on the island that he lived on, which have multiple toes. So they actually look more like that. So the world record for the maximum number of toes found on a cat, and I've looked this up, is 36, which is a lot of toes. How many is that per? Obviously not all on the same paw. What's 36 divided by 4? It's going to be, I'm not very good at math. What's that? 18. Shoot, that's like 9 toes on each paw. They're super cute. I always love doing this. So this is actually one of my students from the summer, I think. A cat called Mr. Tuxedo. which as you can see is not super happy about having his pores photographed. But best thing to do is type in polydactyl kittens. See, I've even got it saved as a search. Shows you how often I look at polydactyl kittens. Isn't that awesome? They're so cute. I wish I had a cat with polydactyly. Unfortunately, all my cats have the normal number of toes. But look at that. They are absolutely amazing at catching stuff too, because they basically have like these baseball catcher mitts of paws. Look at that kitten. Kitten, 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 kitten. Very cute. I could spend ages. No way, Mika. That is super cool. If you can send, if anybody has polydactyl cats, I would love to see pictures of them. We'll make a little collage of polydactyl cats for genetics. It'd be super cool. It's one of my favorite things to talk about. I actually had a friend who had several polydactyl cats, but he never let me take pictures of them because he thought I was going to hurt him somehow. I don't know. He was a bit of a weird guy. Anyway, let's get back to business. Sorry, a little bit of a transgression there. Digression. Yeah, digression. So all of that comes together to initiate transcription in eukaryotes. And it all has to work because otherwise you don't get transcription, right? Or if other things are supposed to stop transcription, you get transcription where you're not supposed to, and you end up with super cool, cute kittens that have more toes than they should do. Can't imagine seeing a cat with 9 toes on each paw. Be very good for walking on snow. Be like wearing snowshoes. Anyway, so as was said before, the actual process of transcription is like ho-hum, you know, whatever. But we've got to stop it at some point because this is a costly business and we don't want to transcribe our entire genome because that'd just be a complete waste of energy, right? Just generate these huge things that we don't need. So we have to terminate transcription somehow. Get back. And it's best understood in bacteria, again, because bacteria are less variant and simpler to study, essentially. And so in bacteria, you have 2 types of transcription termination. Both of them require essentially on 2 different things. Well, let me just change that. Relies on 2 things. 1, slowing RNA pol by long strings of single bases. And we'll get to what that kind of means in a little bit. Basically, RNA polymerase, like DNA polymerase actually, doesn't like seeing lots of the same thing because it has a bit of a conniption about it. Just stutters and pauses and doesn't work very well, right? Likes a varied diet of bases, I guess. And the second is the formation of hairpin. They're called hairpin. They're also called stem loop structures. So those 2 things are central to how both of these work. And so both intrinsic and row-dependent termination both rely on essentially stuff encoded by the transcript itself. You know, and actually like sequence features, essentially. And once those features are encountered, then depending on the mechanism, transcription is terminated. Right, so you'll find these features at the end of a gene. Now, intrinsic is, as you'd guess, something relies only on those features. Row-dependent termination requires those features and a protein, right, to come along and finish the job. And so essentially what we have, and you remember these from transposons. So where were we at? Here we go. So these inverted terminal repeats in transposons are recognized by transposase, right? They're involved in kind of picking out a transposon and sticking it in somewhere else. However, in expression in bacteria, these are able to bind to themselves, right? So if you remember those RNA structures where you had those loops and stems and bubbles and all kinds of funky stuff, right? Essentially, these are kind of opposites, right? And so once they're transcribed, right, so the RNA polymerases come along, you've got a bit of RNA. This is something really important that we'll come back to later. That RNA can now kind of bend and stick to itself and make a little bit of double-stranded RNA, right, that looks like so. This is why it's called a hairpin loop, because it looks a little bit like the loop at the end of, you know, the hairpins that if you had more hair than I do, you'd stick in your hair. So that's one feature. This is actually the one that terminates transcription. The one that slows RNA pol down, right? Because essentially RNA polymerase is going along the DNA. So even if you form these structures and the RNA pol has already disappeared off into the distance, it's not going to get stopped. So you have to slow down RNA polymerase. It goes, train stopping at the station. And only when it's stopped, and it basically pauses on these A's, because it makes this poly-U sequence, stays there, and this inverted repeat here forms into a hairpin loop, and this forms inside the RNA polymerase. So basically that hairpin loop forms in here. And there's not a whole lot of space in there, right? And also because this is a conglomeration of proteins, right, which are all held together by, you know, different interactions and bonds and the like. When you have something bigger than it should be in there, that just forces those proteins apart and the whole complex falls to pieces, right? Because there's not a lot sticking it together, right? It kind of fits together like a, you know, like a puzzle, 3D puzzle, but it's not glued together. It's just held together by its shape. And so when that hairpin loop forms, this thing here, and it forms within that channel, it forces the RNA polymerase holoenzyme apart and transcription stops. So that's intrinsic termination. Row-dependent termination is similar in that it has a terminate sequence, a poly-U sequence. It also needs a stretch of RNA in the transcript which doesn't have any structures, right? Because that is where Rho, the enzyme, binds to, right? And so there's actually a couple of different models, as it, always, right? This is called the torpedo model. And essentially what happens is RNA polymerase is busy chuntering along, and there's this clear bit of RNA, right? No stem loops, nothing crazy in there that Rho can bind to. And once it does, it starts running after the RNA polymerase. And so if the RNA polymerase doesn't get paused, Rho is going to be constantly chasing after it. I don't know, like Tom and Jerry, I guess. However, this terminator sequence is synthesized, and that just slows down RNA polymerase and allows Rho to come barreling in and just go blow it all apart. Kind of. It's not actually an explosion, obviously, but it's similar. And essentially, Rho unwinds this little DNA RNA hybrid, right? This bit here, and results in the RNA polymerase falling off the DNA. So Rho actually physically gets, so it's coming kind of down here from the bit of RNA, and it forces its way into this exit point, unwinds this RNA DNA hybrid, and essentially transcription stops as a result. Okay, anybody got questions? I'll have another piece of chocolate. It's really good chocolate. Hint, hint. Wink, wink. So again, remember the central principles here. Loop formation, slowing down RNA polymerase. Those are the two kind of key characteristics of transcription termination in bacteria. Now, in eukaryotes, obviously more complicated, less well defined as well, right? So this is one way this happens, but there are potentially other ways. And this also ties into what happens to RNA in eukaryotes. So in eukaryotes, RNA has to get processed. There's a lot of stuff that's done to it. Right, so there isn't an inherent termination sequence in eukaryotes. Right, so RNA polymerase, unless it's told otherwise, will pretty much continue forever and ever and ever. Right. However, this pre-mRNA, it's called a pre-mRNA because it hasn't been finished yet. So in eukaryotes have pre-mRNA, and that's an mRNA molecule, that hasn't been processed yet. So it's not available for translation. I mean, it hasn't even left the nucleus yet either, right? But part of its processing is being cut and then having stuff added to the 3' end. Actually, it's the, we go all the way back up here. This poly A signal, or poly A tail, is what gets added after that cleavage point, right? So it's all kind of tied together. RNA processing and RNA transcription of termination of transcription in eukaryotes are tied together, right? So we need to add this on, and to do so, we need to cut that pre-mRNA. There we go, right? So the thing with RNA as well is if you have like a free end flapping around, it's yummy stuff to RNases. So there are a whole ton of RNases in the cell, which as soon as they see a free end of mRNA will come along and they'll just start Pac-Maning it up and chewing it up from the end. And that's really how transcription termination works in eukaryotes. So this cleavage site has a specific DNA sequence, a very specific one, because you don't want this to happen anywhere else. And an enzyme will come in and it will cut it, right? This pre-mRNA now gets processed somewhere else, ready for export into cytoplasm and translation. Now we have a free 5' end, and that's what an enzyme called RAT1 binds to. And essentially what RAT1 does is it chomps up that mRNA as it's being made by RNA polymerase. So again, RNA polymerase isn't going to, stop, but essentially RAT1 then tries to catch up, right? And as soon as it does catch up, and it's again not entirely known how this terminates transcription, RAT1 kind of forces the RNA polymerase apart and it falls off the gene. So in both prokaryotes and eukaryotes, transcription continues past the end of the coding sequence. Right, there's an overrun, essentially. And so that overrun is kind of inevitable, but you also don't want it to be any longer than necessary because that's wasteful. Essentially, this mRNA will get, or this little fragment of RNA just gets chewed up back into nucleotides and recycled. Anybody have questions? Questions so far? Okay. Go on. So now, we can start very briefly on the next section. And so this initial part, processing, is solely a eukaryotic thing, right? And it's worth pointing that out. And there's basically three things that need to be done, right? One, introns have to be removed. This is a process called splicing. Right, so these intervals, these non-coding bits of RNA have to get gone because they're not translated, right? So you can't leave them there because otherwise actually really bad stuff happens many different ways, right? So you got to get shot of these things. Second, the 5' and 3' ends of the mRNA have to be protected. Five prime by a cap and 3' by the poly-A tail there. right? So that feature that you can see here, this helps protect the mRNA. And so that's really important in mRNA stability and also important in translation too. Yes, it does. So basically, poly-A tail is bound by, you guessed it, poly-A binding proteins. Which protect the 3' end from RNAs, particularly exonucleases. So remember I said, where were we? Down here, right? Every time you have a free end of RNA, and RNA is inherently unstable, right? It disappears as soon as you look at it, basically. And so you have to make these ends not look like RNA. So the RNase is like, nope, not interested. And they move on to something else. So that's one of the important roles of the poly-A tail. Did that answer your question? Cool. And then the third thing that has to happen to mRNA in eukaryotes is it has to be exported to the cytoplasm. And so if you look at here, we don't really see the processing, much of the processing going on because it's somewhat simplified. But you can see that the transcription occurs in the nucleus, translation occurs in the cytoplasm. So you have to get that processed mRNA out of the nucleus via these holes called nuclear pores into the cytoplasm where it can be translated. And so the very first thing that happens is literally as soon as the 5' end emerges from the RNA polymerase. Where's that picture gone again? Right, so as soon as this 5' end peeks out of the RNA polymerase, like a little worm coming out of the burrow, it has to be capped. Because otherwise, an RNase will come along and go, ooh, tasty, and would start degrading it as it's being produced. And that will just be plain annoying, right? Because then the RNA polymerase will be just making something to be degraded. So as soon as that 5' end is emerges from the RNA polymerase, it has to have a cap added to it. And that's called the 5' cap or the 7-mpg cap. And that's short for methyl, come on, guanosine. I can spell it right. I cannot. No, that's not any better. Bloody hell. And essentially that methylguanosine, if we kind of pop down here, looks nothing like RNA. So once this funky ass kind of structure, which is just a whole bunch of kind of sort of RNA nucleotides with, you know, methyl groups added to them and all kinds of funky stuff. When that's added, now that growing 5' end is invisible, essentially, to the RNases. That 5', oops, there we go, cap is also really important in translation, because it's where the small ribosomal subunit attaches. And we'll get into that in more detail in the section on translation. So, and I'll probably just finish going through these 3 slides, because these are basically, you know, a pictorial representation of what I just put in the chat. So as that transcript is being transcribed, right, first you cap the transcript, right? Literally as soon as it emerges from the RNA polymerase and it's accessible to the various capping proteins. Then as that transcript is made, RNA polymerase is, you know, essentially over here, going that away to the right. Now you produce an intron, the first intron. Now we recruit the splicing machinery, right? So this will recognize and remove these introns, and we'll go into that in some depth Thursday. Transcript keeps getting made, right? So, you know, these introns are going to get spliced out. Actually, somewhat inappropriate, but I always think of Brad Pitt in Fight Club. Don't know if anyone's ever watched that film, when he splices rude pictures into kids' films. Anyway, it's the same, you know, splicing is actually something that's used in filmmaking as well. But we're just talking about RNAs here. If you don't know what I'm talking about, probably you're better off not knowing, but it's a really cool film. It's also a really good book as well. But anyway, this process continues, not necessarily in the order in which the introns are produced, but they all have to be removed at some point. And then the very last part is the adding of the poly-A tail. So it's polyadenylation. It's poly-A because there's just a string of A's after it. And so what you end up with is what's called the mature transcript. So the mature transcript has 5' cap, 3' poly-A tail, and no introns. It is one continuous protein coding region in between, from the translation start site to translation stop site. Obviously, there will still be the 5' and 3' UTRs, but there will be no introns present. Once all of this stuff has happened, it looks like this, you know, diagrammatically, then it can be exported to the cytoplasm. Because as soon as it gets down to cytoplasm, then it can be translated. So you got to do all this stuff first. You can't wait until you're in the cytoplasm. Ta-da! Okay, so next class we'll get into splicing and SNRPs, which is my favorite acronym in molecular biology. Cool. I'm going to have something to eat before lab. So I'm going to say goodbye for today, and I will see you either in 14 minutes or on Thursday. So have a great day. See you soon.